The presence of unrepaired DNA double-strand breaks could be disastrous for mitotic cells because they result in the formation of chromosome fragments that lack centromeres, and which are therefore unable to attach to spindle microtubules via kinetochores. This could result in chromosome missegregation and ultimately lead to the development of cancer. Surprisingly, however, acentric chromosome fragments are somehow able to segregate to the spindle poles in cells from a variety of different species. Bill Sullivan's lab at the University of California, Santa Cruz, for example, previously observed the successful segregation of acentric chromosomes generated by the expression of an endonuclease in Drosophila neuroblasts.
Postdoc in my lab, Ann Royal, saw that when we made an acentric, they would segregate later than all the rest of the chromosomes, but pretty well. Sister acentrics would separate and go to opposite poles. And because they segregate fine, these flies survive. And discovered this strand of DNA that seemed to be pulling them towards the poles. You know, if you have an unrepaired double-stranded break and you're in metaphase and about to go in anaphase, that's like a worst-case scenario for a cell. And so we viewed it as a last Hail Mary that you can segregate these acentrics with this DNA tether and they can be repaired in the next division.
The DNA tether links the broken end of the acentric chromosome to the main chromosome mass. Following up on the previous study, Sullivan and colleagues, led by graduate student Travis Karg, reasoned that, if the tether really does pull acentric chromosomes to the spindle poles, the acentric's telomere should always trail the rest of the chromosome fragment as it travels polewards.
Travis started doing movies where he looked at the telomere, and we realized that they were segregating either telomere first or telomere last 50% of the time. So that made us rethink whether the tether always provided the force or were there additional mechanisms.
Previous studies have suggested that acentric chromosomes associate with microtubules, so Karg et al. Simultaneously imaged acentric chromosome segregation and microtubule dynamics. Acentric fragments appeared to associate laterally with interpolar microtubules, that is, microtubules that extend from centrosomes towards the cell equator that aren't bound to kinetochores. In particular, segregating acentrics associated with interpolar microtubules at the outer edge of the spindle.
So then we had an idea that it was specifically these interpolar microtubules that were involved in transporting the acentrics. And then to really experimentally demonstrate this, we got together with Sophie Dumont's lab at UCSF, and she has this wonderful laser ablation system where you can ablate microtubules. And what we could really see when we ablated the microtubules between the segregating chromosome and the normal mass of chromosomes at anaphase, the acentric stopped moving. And then the microtubules reestablished themselves, build up a new set. And as soon as that new set was built up, they would start segregating again. So it seemed pretty clear they were dependent on microtubules.
In contrast, acentric chromosomes continued to move polewards when the researchers laser ablated the DNA tether linking them to the main chromosome mass. The tether is required for the segregation of acentric chromosomes, however, because mutations in components of the tether, such as BubR1 or polokinase, inhibit acentric segregation and cause the death of flies expressing the acentric generating nuclease ICRE1. To identify additional factors required for acentric segregation, Karg et al. Screened for genes that show synthetic lethality with ICRE1 expression.
So Travis went through some 100 candidate proteins, and one of them that came out really strong was this chromokinesin KLP3A.
Chromokinesins are motor proteins that bind to both microtubules and chromatin. KLP3A mutant flies expressing ICRE1 in their neuroblasts show defects in acentric chromosome segregation and decreased rates of survival. But KLP3A is considered to be a plus-end directed motor, making it unlikely to be directly involved in the -n-directed transport of acentric chromosomes to spindle poles.
And so going through this, what we realized is actually, and this was also in the literature too, the other defect, if you look at spindles in a KLP3 mutant, at least in Drosophila, they form a spindle. They're a little bit shorter, but they don't make these outer interpolar microtubules.
Acentric chromosomes usually segregate along these peripheral microtubules, but in KLP3A mutant neuroblasts, acentric chromosomes were positioned in the spindle interior and failed to move to the spindle poles.
And so what we're thinking now is that KLP3 is necessary for making these interpolar microtubules, and acentrics rely on those to segregate. And one thing I like about this story, you think about it, you have an acentric fragment that's really recombinogenic, so you want to keep it away from the other chromosomes. And so the two things you do is, one, it segregates much later than the normal chromosomes, so it's temporarily separated from the normal chromosome segregation. But now it uses these interpolar microtubules way off to the side. So one idea is that this is just the cell's way of keeping this dangerous chromosome with a broken end away from all the other chromosomes.
Karg et al. Think that the DNA tether may provide some of the force for moving acentric chromosomes along interpolar microtubules, but they still want to identify a -n-directed motor protein that can mediate this process. The tether may also prevent acentrics from drifting too far away from the metaphase plate, and after segregation, it delays nuclear envelope reassembly so that the broken chromosomes can be incorporated into daughter cell nuclei. Sullivan and colleagues are also interested in what happens when this latter process fails and acentric chromosomes are instead incorporated into their own separate micronuclei. For now, though, you can learn more about how the chromokinesin KLP3A and interpolar microtubules facilitate acentric chromosome segregation in the paper by Karg et al., published in the June 5, 2017 issue of the Journal of Cell Biology.